Hi, Michelle Glass here. We are ready to continue our conversation of photosynthesis here in another one of our Chapter 7, Lecture 7 episodes. The topic for this video is the Calvin cycle, or we can think of this as our sugar factory. This is where we will actually be making our sugar molecule. Now this process, Calvin cycle, is taking place in the stroma of the chloroplast. So that is the fluid component, remember, surrounding the grana. This is the step where we're going to take our carbon dioxide gas from our atmosphere and fix it, meaning turn it from an inorganic carbon dioxide molecule to an organic glucose molecule. This process is going to require the NADPH and the ATP that we produced as part of the light reaction. Of course, we didn't do that, but the plant did. And then this will recycle the NADP plus and the ADP endophosphate group that are necessary to continue another round of the light reaction. Remember, carbon fixation is our term that's talking about taking an inorganic carbon from carbon dioxide gas and turning it into an organic molecule, meaning that other cells can now access and use it. We are still in the chloroplast, so here's just our reminder of our organelle, and our stroma is that fluid surrounding those stacks of thylakoids. Okay, so to begin, you're going to have a molecule called RUBP, which is going to pair with carbon dioxide gas. You have to have the enzyme ribisco in order to convert it into a 3-PG molecule. So we need to pay attention to this enzyme ribisco here because it's going to play a role in a later video presentation. This is actually our carbon fixation step where we are combining carbon dioxide gas with RUBP to make a 3-PG molecule. From there, we really just are producing our G3P, which is going to ultimately become our sugar. So our next process is going to be requiring the ATP and the NADPH that we made as part of our light reaction. Notice that we are going to be forming ADP and phosphate and then NADP plus as part of this. And then we need to regenerate that RUBP so that the cycle can continue to occur. And so that's what's going to happen in our final step. In each one of these steps, there's lots of different molecules that are formed, lots of different enzymes that are involved. We are not required to know all of those details. Notice in the regeneration of RUBP, we have ATP being used, and of course, another ADP and phosphate being formed. Notice that all of the ADP and phosphate groups as well as NADP plus then becomes available for the chloroplast for the light reaction. So we can just continue these two processes together. Even though we think of the Calvin cycle as the dark reaction or the independent light reaction, it is still requiring the energy from the light reaction. And so what we typically see happening is that these reactions are happening simultaneously. So think of them as happening together side by side. That G3P molecule is going to be converted in the cell into our glucose molecule that we've been talking about. Glucose can then be used by the plant cell at the mitochondria for cellular respiration, which is, again, our process that's used by plants and animals to produce ATP molecule. The glucose can also be linked together to be stored as a starch molecule, which is a polysaccharide, or it can be linked together to make cellulose, which is going to be important in the plant cell wall, or you can use your G3P to make other organic compounds such as proteins and lipids and so forth that the cell needs. All right, that is it, but stay tuned for a final discussion on how plants adapt to different kind of weather conditions. How are they able to continue doing photosynthesis even when it's too hot for those stomata? Remember those holes in the underside of the leaves to be open. So stay tuned, but that is it for now.